This is the Onco'Zine Brief with Peter Hofland and Sonia Portillo.
Today in the Oncology Brief, we talk with Charli Johnson, Chief Executive Officer of ADC Bio, a biotechnology company specialized in the development of antibody-drug conjugates. Antibody-drug conjugates, also known as ADCs, are a novel type of targeted anticancer agent. Today, 4 drugs in this class of agents have been approved by regulators around the world. I'm Peter Hofland, Sonia Portillo is on assignment, and this is the Oncology Brief. The antibody used in an antibody-drug conjugate, or ADC, is specific to so-called cell surface proteins on the surface of cancer cells. In simple terms, this means that the antibody is able to specifically target cancer cells. This approach offers various advantages over current therapies based on standard chemotherapeutic drugs currently used in the treatment of cancer. One of the benefits is that ADCs are designed to only kill cancer cells, but do not harm healthy cells. In today's episode of the Oncology Brief, we talk about the manufacturing of ADCs, including the complexities of the typical supply chain. In some cases, the typical supply chain may involve up to 5 so-called contract development and manufacturing organizations, or CDMOs. The reason of so many parts in a manufacturing process is that the development and manufacturing of ADCs is really complex and requires specialized companies. So, what is involved? The manufacturing process of an ADC involves the development and manufacturing of a potent cytotoxic payload, and this is the actual cancer-killing drug. A biodegradable linker, monoclonal antibody, the conjugation of the payload via the biodegradable linker to the monoclonal antibody, and finally the fill-finish process and packaging for distribution. And each element in the manufacturing process is treated as a unique, separate activity. Adding to the complexity is that each step of the process typically involves shipments and transfer of material between different CDMOs. This may, in many cases, mean shipment across international borders. For example, it is not unheard of that an antibody is manufactured in one country in Europe, but that the conjugation with the combined linker-drug part takes place in a facility in another country. As a result, the challenges for biopharmaceutical companies developing ADCs is that this approach involves a high degree of redundancy, risk, cost, and time. Economic analysis suggests that the delivery of multiple elements of the supply chain from a single source location could save months in the development and time of these novel therapeutics. The analysis also says that in some cases, this may be up to 6 months for a total manufacturing process. And this is important, because in simple terms, this helps speed up the development of an ADC, and ultimately makes it possible to get very powerful anti-cancer drugs in the hands of clinicians to treat their patients faster. And ADC Bio plays an important role in the development and manufacturing process of novel therapeutics. The company develops new process technology designed to speed up, simplify, and significantly lower the production cost of the latest generation of these anti-cancer blockbuster drugs. In our interview, held during CPHI Worldwide in Madrid, Spain, we talk with Charli Johnson about antibody-drug conjugates and the role his company plays in the development and manufacturing process. We also talk about the complexities and trends in the development of other novel medicines, from large bulk manufacturing to hyper-personalized medicines. And finally, we briefly talk about financing of companies like ADC Bio. CPHI Worldwide is an annual event which hosts more than 45,000 visiting pharma professionals over a 3-day period. During the event, more than 2,500 exhibitors from over 150 countries show what they can offer drug developers and drug manufacturers. And every sector of the pharmaceutical market is represented under one roof. Last year, this event took place in Madrid, Spain. Let's listen to the interview. We're here at the 2018 edition of CPHI, the Pharmaceutical Industry Market. Here with Charli Johnson, Chief Executive Officer of ADC Bio, ADC Biotechnology is a CDMO with a lot of innovation in the back pocket, and actually getting a little bit more right now for people to know what's going on with the potential of creating better, faster, more interesting ADCs. This year has been a very exciting part of the company, but before we're going to talk about the company, welcome to the show, Charli.
Thank you, Peter. Thanks for the opportunity.
As I said, you're a CDMO. There's a lot of things that happen in CDMOs. CDMOs are contract development and manufacturing organizations. Tell me a little bit about the changing nature of CDMOs.
Sure. So we're a specialist CDMO. We develop and manufacture ADCs, and that's all we do. So we're not a classical CDMO in the respect of some of the bigger players that do all sorts of other things with small molecules and biopharmaceutical manufacturing. But yeah, we are one of a few specialists in the area of ADCs, and ADCs are an interesting challenge. They combine, skills in manufacturing of small molecule payloads and biopharmaceuticals, antibodies and proteins and the like of. So that's what we do, and that's what our company does, and what it was set up to do. We started from the background of improving ADC development and manufacturing through technology. We continue to do that, and we're moving in to become a mainstream player in terms of manufacturing of clinical trial materials.
So, if I understand correctly, the D in CDMO, the development part, it's a major part of your company.
It's been incredibly important to us, actually. We have now between 30 and 35 clients that we service regularly. We turn over in excess of 50, 60 projects a year in the R&D space, and the development piece is exceptionally important. We're lucky, actually, that we've had our hands on lots of the different technologies, linker technologies, payload technologies, and enzymatic technologies that ADCs rely on these days. So we have a very, very broad experience in the R&D sector of ADCs, and we're looking to deploy that into manufacturing at the behest, actually, of our clients and our investors. They see this as a very attractive and vibrant market.
Now, if you look at your company right now, it's been, what I said earlier, it's a very exciting year for you. You were able to secure some additional financing. Tell me what that means for you as a company.
Sure. So working off the back of the investment that we took in September of 2017, which was primarily to build out our new facilities in North Wales at Deeside, which is effectively to consolidate our existing R&D services alongside clinical manufacturing of conjugates, of ADCs and bispecifics and the like of. We took in some top-up funding in April of this year, essentially to do a couple of things. One was to bolster our business development capabilities in continental North America. And the second thing was, in response to industry trend, was to look at conceptually designing a fill-finish capability within the Deeside footprint. So we're currently occupying around about 50% of the footprint, and we're now looking to go downstream into fill and finish.
That means that you are right now able to produce ADCs from early beginnings all the way till a finished product.
Not exactly right now. I mean, we can do that in an R&D setting, and we have done for many years. But the intention is by the first quarter of next year to be capable of producing clinical materials for bulk ADCs, and thereafter, within the next 12 to 18 months, to be able to follow that down into formulation, fill and finish, because the industry is asking for that, and they want to consolidate those activities.
Yeah. Now, let's take a short break. If you're just joining us, today in the Oncozine Brief, we talk with Charli Johnson, Chief Executive Officer of ADC Bio, a biotechnology company specialized in the development of antibody-drug conjugates. Our interview was recorded during the 2018 edition of CPSI Worldwide, held October 9 through 11, 2018 in Madrid, Spain. I'm Peter Hofland, and this is the Oncozine Brief.
Each day, researchers make discoveries that bring us closer to the moment when all cancer patients can become survivors. Their progress is made possible with the help of clinical trials. Clinical trials are the brightest torch researchers have to light their way towards better treatments. And if you've been diagnosed with cancer, they may be your brightest ray of hope. Speak with your doctor and visit standuptocancer.org/clinical trials to learn more. Together, we can stand up for all of us.
This is the Onco'Zine Brief with Peter Hofland and Sonia Portillo.
And welcome back. I'm Peter Hofland, and this is the Onco'Zine Brief. If you're just joining us, today in the Augustine Brief, we talk with Charli Johnson, Chief Executive Officer of ADC Bio, a biotechnology company specialized in the development of antibody-drug conjugates. Our interview was recorded during CPHI Worldwide, held October 9 to 11, 2018 in Madrid, Spain. Now, you are... Earlier on, we were talking about the development part of it and the innovation that you're trying to bring to the ADC world. One of the things that you were talking about earlier this year, different presentation, was a new upstream bioconjugation process. Tell me a little bit about that.
Yeah, sure. And I think this is an interesting paradigm shift. So if you go back sort of 10, 15 years, the classical way of developing an ADC was to manufacture an antibody and test an antibody in the first instance, and then look at a follow-on with an ADC. And so classically the way to manufacture is you develop and produce an ADC, an antibody, and then you would use that as a starting point for then developing an ADC and selecting a payload, selecting a linker technology, and the ADC is then the follow-on product. The way that the industry is developing right now is that people are just interested in skipping that first part and moving directly to an ADC.
It means skipping, meaning not we're looking at the monoclonal antibody first, but really looking at what the ADC can be.
As the primary product, yes, exactly that. So what we've conceptualized is, based upon our core background technology lock release, is can we sort of intervene at an earlier stage? So rather than isolating a monoclonal and purifying it, and the normal way of things is, can we enter the process earlier and use one of our resins to actually sequester the antibody from a crude soup solution of the antibody, carry out the conjugation, and then carry out the downstream purification, hence saving time, saving costs in terms of protein resins, and also consolidating the analytical burden related to a separate monoclonal step and then a separate ADC.
You said saving cost. Tell me about that.
Yes, so one of the most expensive parts of an antibody process is the protein A resin that is used to isolate the antibody from the crude supernatant. We can replace or potentially replace the protein A by using one of our proprietary mimetic resins. And because it is a mimetic, it's not proteinaceous in nature, we can then directly go to the conjugation step, produce the ADC, and then carry out the downstream purifications and viral inactivation and clearance on the ADC itself, rather than having to do that in a pre-step with the antibody.
Now, you said you based this on your lock and release technology. In short, because some of our listeners may not be familiar with a lock and release technology, which is different than being used by other companies. Real briefly, can you explain a little bit about the technology and why this is so important? I mean, one of the things that was presented earlier this year, it also helps avoid aggregation or limits aggregation. So there are certain benefits to the technology that we've not seen in other ways of developing ADCs.
That's right. And we've been using, we developed lock release technology nearly 8 years ago now. And we've been using that certainly very predominantly over the last 5 years to aid people who are in the R&D phases. And lock release helps early innovators to produce ADCs very quickly and in a very clean format. So a format where there is limited amounts of residuals, and particularly the payload, so that you get a very sort of clean readout in the initial in vitro panels. And then subsequently, we've used it to avoid aggregation, as you say. A lot of the newer payloads, the DNA alkylating payloads, for example, are highly hydrophobic in nature. They have a high propensity for aggregation when you make the ADC. Using the lock release technology, which effectively segregates the antibodies at a very early part in the processing, allows you to produce highly monomeric and clean conjugates when you release them from the resin, and you get a very clean product which is free of residuals, high yielding as well. So you avoid maybe 20-30% aggregate that you have to remove in subsequent chromatography steps. So there's a yield benefit, a quality benefit, and a speed to market benefit of using this technology.
Which is obviously very important for your clients.
Indeed it is, yeah.
Now, one of the presentations here at CPHI, which was remarkable, is the enormous growth of the ADC market. If you look at today, there are 4 ADCs on the market, at least commercially approved in the United States, and I guess also around the rest of the world. In the rest of the world, there are a whole bunch of ADCs right now in late stage phase 3 clinical trials. What are your expectations if you look at this market right now of ADCs in the next 3 to 5 years?
I think, I'm usually healthily skeptical about the research that comes out around, how quickly these things are going to commercialize and grow. But I think in the case of ADCs, we're actually not far off the predictions that have been made for the last 5-10 years, that, we're going to be looking at an overall market of between $15 and $20 billion before too long. And, that's on the basis of the growth of the 4 commercially launched ADCs, but ones that are in later phase development. And certainly where we're at right now, which is servicing early clinical and mid sort of clinical manufacturing services, we're seeing a growth rate of 50+% this year in 2018 from last year, and we're over 85 ADCs, individual ADCs in clinical development right now. It's a rapidly growing market. There was a little stutter back in 2015-16, but it's recovering incredibly strongly. And I think that in combination with the checkpoint modulators is seeing a huge potential for ADCs, not only as single agents but in combination as well.
Now, the majority of ADCs are still in oncology or hematology. One of the exciting things is to see that I think about 14-15% of development of ADCs are now taking ADCs outside of the world of oncology and hematology. But that comes with its own problems. Can you share with us some of the things that may be considered a problem when you take an ADC from an oncology-based product to an outside non-oncology product?
Yeah, and we've worked in this area on a couple of occasions. So we've worked with steroidal drugs in combination with antibodies, and without saying too much, we're starting to work with companies that are looking at antiviral products as well. And really, from a technology perspective, the challenges are similar. The linker technologies are quite often different, but when it actually comes to the development and manufacturing challenges, the things that we see are fairly similar to what we experience with the oncology products. So again, hydrophobicity of payloads can make it difficult to, produce formats which, you can utilize. Linker technologies are interesting as well. Within our group, we have a chemistry function as well, which, customizes payloads and linker combinations, customizes release mechanisms so that you can access, when the ADC finds its target and is internalized, it can release its payload in a format that is therapeutically beneficial and does its job. And so the linker and the release mechanisms associated with that are incredibly important. So it's important for any ADC specialist to have not only the ability for the bioconjugation, but also to be able to tailor the chemistry side of linker and payload.
Let's take a short break here, and then we talk some more with Charli Johnson, Chief Executive Officer of ADC Bio, a biotechnology company specialized in the development of antibody-drug conjugates, recorded during the 2018 edition of CPHI Worldwide, held October 9 to 11, 2018, in Madrid, Spain.
Some of the best sounds you'll ever hear are generic, safe, effective, even money-saving, just like FDA-approved generic drugs. Even if they don't come in the exact same color or shape as their brand name equivalents, they have the same key ingredients and go through a rigorous review process. Talk to your doctor or pharmacist today and visit fda.gov/generic drugs. Generics are safe, effective, and can save you money. You'll like the sound of that.
This is the Onco'Zine Brief with Peter Hofland and Sonia Portillo.
Welcome back. I'm Peter Hofland, and this is the Onco'Zine Brief. If you're just joining us, today in the Onco'Zine Brief, we talk with Charli Johnson, Chief Executive Officer of ADC Bio, a biotechnology company specialized in the development of antibody-drug conjugates, recorded during CPHI Worldwide, held October 9 to 11, 2018, in Madrid, Spain. Now, one of the exciting things that's also being observed here at CPHI, but also beyond CPHI, is the fact that, and I think it's a challenge in itself, that some companies are actually thinking about placing two different kind of payloads on an ADC.
Yeah.
I think it's a very exciting thing to see, but I think from a manufacturing side, the development side, maybe the linker chemistry, that might be a nightmare.
Yeah, and I mean, it's already complex. Making ADCs is already complex, and then, accessing sufficient orthogonality in terms of linkage sites and for different payloads is an interesting challenge in its own right. It's not something that we have worked with so far, but this idea has been around for some time, and I think there's a lot of promise in terms of, certainly in the oncology space, conjugating payloads which have complementary modes of action. It's going to be an interesting challenge, I think, from a chemistry perspective as much as anything else.
Those very novel drugs are not to be expected for a little while, I assume.
I think they're in R&D right now. I mean, it's a compelling concept at this point, and we'll see how that develops.
Now, talk about the economy of ADCs. The economic data is showing some very interesting trends. Your company's been doing some of that, and without going into all the details with numbers, can you share with us some of the interests or some of the interesting economic data that you've noticed, observed, or gathered?
Yeah, we can do that. I think this fits broadly into two aspects. So supply chain is one thing. One of the things that we are increasingly asked by our existing clients and new clients alike is, can we do more? Can we either do the antibody part and the conjugation? Can we either do the conjugation and the fill and finish? Because they see huge benefits in terms of time to clinic, in terms of combining those things together. So that's one thing. That's a time saving for clients, and there are also cost savings associated with release, quality control, et cetera, et cetera. The other thing that we're looking into is being able, as I mentioned earlier, to combine the conjugation event earlier in the upstream manufacturing of the antibody. We see that we can save up to 50% of manufacturing time there. We see that we can save somewhere in a region of 25-30% in terms of costs. That comes from savings in time, but it also comes from savings in material consumption, usage, and also the protein A resins. So we're really excited by that. We see that as a paradigm change in the way that ADCs are developed and manufactured, and we're really excited about that. In fact, we're starting to work with some of our existing clients collaboratively to make those things a reality.
Now, CPHI is not only about ADCs, and you walk around here, and I assume that you see also some of the other trends being presented. One of them, of course, if you look at very novel and very personalized medicine technology, like CAR-T, chimeric antigen receptor T-cell technology, it's very, very, very personalized, and basically based on the individual patient, the drug is being manufactured. On the other side, you see the increase in bulk manufacturing in single-use technology, where right now people are talking about 3,000 to 4,000 liter bioreactors. From where you are observing the industry, there's in that respect a two-way split. But what are some of the trends that you see are happening in medicine? Is it more a move towards the hyper-personalization of medication, or is that still the bulk manufacturing of, for example, ADCs, but also other drugs that may be manufactured in that way?
Yeah, that's it's certainly an interesting sort of trend that's going on there, and I think, I think you describe it correctly. Hyper individualization of medicines. It's still, it's a very compelling concept. ADCs have often been sort of branded as personalized medicine. I mean, they're personalized antigens, to specific antigens, and I think that sometimes gets a little bit lost. I think we're some way off that real personalized medicine becoming a reality, but it's a concept that has been talked about for a long time, and I think it's progressing. If I look a little bit sort of closer to home, one of the key things that we see emerging in terms of biosimilars is, we're having a lot of conversations recently with biosimilar companies, Asia, India, looking at ADCs very keenly now, and looking to enter that space. The patent lifetime for some of the bigger products like Kadcyla, are coming close, and there's an awful lot of interest being generated there. Likewise, innovative ADCs from China. We attended the inaugural event, World ADC, over there earlier in the year, and there is a huge amount of activity, a lot of innovation, as well as Me Too products coming. You mentioned about the fact that some of the products, the first and second generation ADCs, are now getting closer to their end of their patents in Western Europe, around the world. Yeah. What does that mean beyond the fact that now companies may look at the potential for creating a biosimilar? Yeah, I think it's interesting. We've had lots of conversations recently, particularly around Kadcyla, actually, and the discussion always starts with: do we want to go down a true sort of biosimilar route, or do we want to be bio-better? And looking at alternative payloads. And I think most people who are active in that field, they have already a generic trastuzumab. Right. So the easiest entry point really is the biosimilar route with Kadcyla, but we always have the conversation with potential clients about bio-better. Should they go down biosimilar or bio-better route? And it's coming. It won't be long before Adcetris is added to that list. When you talk about bio-better, you see also original trastuzumab-based ADCs that are in development. But it's still based on trastuzumab, on the HER2 problem in that respect. So when you look at the first generation ADCs and you look at the second generation, right now we're talking about the third generation of ADCs. What are some of the key differences that we do now completely different than when we started off ADCs? Yeah, it's another very interesting point. So, I mean, we've moved to site-specific technologies. a lot cleverer technologies in terms of the conjugation event, plasma stability, all of these things, and, to ever increasingly potent payloads. And then I think we've taken a little bit of a pause based upon some of the data that clinical data that we've received recently. And now we're starting to sort of attenuate those things. And it's a gradual process of, optimizing linker technologies, site specificity, payload potency. Those things are all incredibly important, but on a ADC by ADC basis, there isn't a Bible that tells you exactly how you do this, and it's an incredibly fascinating place to watch from, actually, to see how these things are being optimized and developed.
Now, both for your company, you're developing new technologies, new ways of looking at ADCs, making them better. Last year, CPSI, we had a little discussion about some of the things that you were doing. It was very exciting to listen to. This year, you had a lot of very exciting things that came to market, were introduced. What about next year?
Next year is going to be all about filling our new facility. So we're looking towards a regulatory inspection, MHRA, in the first quarter of 2019.
Which is the English version of the FDA.
Correct, that's right. And we've been in dialogue with the regulator throughout our build, and so we're not expecting surprises there. It's going to be about getting through that hurdle, getting first products into our facility. We already have some lined up. Incredibly exciting in that respect. It's also going to be about conceptually designing formulation fill and finish, which is additive to that. And it's going to be about starting this upstream paradigm shift in how we manufacture ADCs. And we'll see whether we get to that.
Will be again an exciting year, I must say.
Indeed, yeah.
Okay. Now let's take a short break. If you're just joining us, today in the Oncozine Brief, we talk with Charli Johnson, Chief Executive Officer of ADC Bio, a biotechnology company specialized in the development of antibody-drug conjugates. Our interview was recorded during the 2018 edition of CPSI Worldwide, held October 9 through 11, 2018 in Madrid, Spain. I'm Peter Hofland, and this is the Oncozine Brief.
Did you know that generic drugs are just as safe and effective as brand name drugs? Generics might look different, but they work the same way. And they can even save you money. Don't believe me? Ask your doctor or pharmacist. Or visit fda.gov/generic drugs.
This is the Oncozine Brief with Peter Hofland and Sonia Portillo.
And welcome back. Our interview today was with Charli Johnson, Chief Executive Officer of ADC Bio. It was originally recorded during CPSI Worldwide, the pharmaceutical industry meeting held in Madrid, Spain. One thing where we don't really talk a lot about is funding. I mean, funding of proprietary technology, funding of biotechnology is often very complex. And a lot of companies or a lot of potential funders are really skeptical about very novel technology. In the case of antibody-drug conjugates, also in the case of your company, things are changing rapidly. Tell me a little bit about the funding landscape in that respect, and maybe in this case primarily about the United Kingdom, but maybe what kind of lessons can be learned from other areas as well.
Sure, yeah, that is an interesting point, and I think the landscape actually is changing, particularly for us. I mean, I think if you go back to the start of our story, we're atypical in terms of the way that we have brought ADC technology to the market. I think in some respects it's quite classical. We were a technology startup. We had a background technology that we were developing, but we've morphed into a more mainstream CDMO service provider as well. Now, typically, when CDMOs enter a new phase of technology like ADCs, they do that from the position of an established business, which has, got internal funds to support that kind of activity. So we're kind of, I won't say unique, but we're kind of atypical in that respect. So what's incredibly important is track record. And we have good track record with our existing investor set. We were primarily funded by Development Bank Wales, as they are now, Finance Wales formerly, and we were supported by Seneca Partners, EIS funds, so tax-efficient funds in the UK from 2011 onwards. When we took in the larger round of investment last year in September, we brought on 2 new VCT funds.
Venture capital funds.
Venture capital funds, yes. So Maven Capital Partners and in April this year, Downing LLP, London-based. And they're very, very good supporters and partners, investment partners for us. And the landscape has changed recently for VCT funds. It's no longer the case that they can invest primarily in more established, stable management buyout type businesses. They are more aligned with the EIS landscape, which is the tax-efficient funding route in the UK. And they have to now invest in more risk-associated businesses. So there's a lot of VCT funding now available for companies like ourselves, technology-based companies, startup companies, where risk capital is classically deployed, and that's very good for us. The key thing is to pick the right partners, those that are in it for the medium term and who can support you as your business develops.
So obviously this is not only good for you in terms of ADC biotechnology, but that landscape is also impacting other companies in the UK in terms of getting financing for maybe some of the challenging products that they may try to develop in either ADCs or similar kind of products.
My read of it is there is an excess of funding and a lack of good technology-based projects for these funds to invest in at the moment.
Well, hopefully we can see in the next year a couple of very exciting products being brought to those VCs.
Indeed.
Well, we say thank you very much for your time.
Thank you, Peter.
This edition of the Oncozine Brief was a co-production with ADC Review, the Journal of Antibody-drug Conjugates. For more information about the journal and background information about ADCs, visit the journal's website at www.adcreview.com. If you want to learn more about personalized medicine and targeted therapies in cancer, including the use of antibody-drug conjugates, visit cancer.net. This is the website from the American Society of Clinical Oncology and includes doctor-approved patient information. The website is www.cancer.net. Another valuable resource is the website from the American Cancer Society. This website offers a wealth of information about cancer, including risk factors, symptoms, how a particular cancer is found, and how it is treated. The website of the American Cancer Society is www.cancer.org. For us here at the Oncozine Brief, we want to thank you, our listeners and underwriters, for your ongoing support. Thanks to your support, our program now has a wider reach with distribution via iHeartRadio, in addition to PRX Public Radio Exchange, and in the United Kingdom and mainland Europe via UK Health Radio. You can also download our program via iTunes. In Arizona, you can listen to the Oncozine Brief via Independent Talk 1100 KFNX, one of the top 10 radio stations in Arizona, reaching almost 5 million people throughout the state. For more information about that, check our online journal Onco'Zine at www.oncozine.com. We know that based on this interview and the additional subject matter we've discussed, you may have questions. So please submit your questions to our editorial team via email, Facebook, or Twitter. And we will post as many answers as we can on our website Onco'Zine.com. That is ONCOZINE.com. To help make this program possible, please visit our page at patreon.com/The Oncozine Brief. That is Patreon, patreon.com/The Oncozine Brief. Your support for this program is important. It allows us to bring you interviews with experts involved in the development of novel diagnostics and new treatments. So please visit our page on patreon.com/The Oncozine Brief. If you're living in the United States and want to receive our newsletter, text the word cancer, CANCER, to 66866, and we will make sure that you'll receive our newsletter, which includes an overview of the latest news in oncology and hematology. Thank you all. And thank you for listening, and join us again for our next episode. I'm Peter Hofland, and this is the Oncozine Brief.
The Oncozine Brief is produced for Sun Valley Communication by Peter Hofland, Sonia Portillo, Evan Wendt, David Kaler, and Sean Mayer, and distributed by InPress Media Group. Support for the Oncozine Brief comes from listeners of this station and our commercial underwriters and advertisers. For more information about underwriting and sponsoring options, contact Sean Mayer in California at 949-923-1660 or visit our website at oncozine.com/underwriting. The Oncozine Brief contains health and medicine-related information and is provided for educational and entertainment purposes only. The content is not intended as a substitute for professional medical or health advice and does not replace your doctor's advice. Your doctor is the best person to answer questions about your personal health. If you hear something in this program that doesn't agree with what your doctor has told you, ask him or her about it.